Okay, so welcome everybody to the Ingham seminar. My name's Michael Barton. I'm one of the investigators on this CaVa project that we're going to talk about today. Uh, Professor Bouquet is not available, so he's asked me to chair today's session. And the session is called The Future is Now for Clinical Cancer Registries. It's going to be a bit of a tag team. I'll start with a little introduction. Then Nazreen Kadan, who's the head of the Clinical Cancer Registry in Southwest Sydney, will continue, followed by Georgie Kennedy and Tim Churches. Here we are, here's the plan, who are going to talk about how we make Mosaic computable and what that actually means. I think Georgie will explain, and look at some of the potential analyses that we can do if we do make Mosaic computable. And then Tim is going to finish with the big picture. And we're all going to keep to this very short time scale. So Georgie, could we go on to the next slide? The main motivation for this project is to reduce unwarranted variation in clinical practice. And if you just leave it on this for a second so I can explain it. This shows the age standardized cancer mortality per 100,000 population for local health districts. And you can see there's quite a variation. If we ignore the Albury group, which is fairly small, and look at Northern Sydney, we're going from under 8 deaths per 100,000 to nearly 10 deaths. So about a 25% increase, just depending on the LHD you happen to be in when your cancer is diagnosed. Now that could be due to late diagnosis, more advanced stage, or differences in the treatments that are received. And that's something that we don't know, but clearly, if we go to the next part of this slide, Georgie, thanks. This has consequences. So this is the excess cancer deaths. If everybody had the same cancer mortality as Northern Sydney, then we would reduce the excess cancer deaths by something like 6,000 cases in New South Wales. So quite a considerable effect of the variation in mortality, and that variation in mortality stems from variations in presentation, diagnosis, and treatment. So if we go to the next slide. This is one of the major themes of the Sphere Cancer Clinical Academic Group, is reducing unwarranted variation in clinical practice. And the first thing we need to be able to do is to measure that variation and then to be able to test what happens. So this project called CaVa combines the clinical cancer electronic medical records, so Mosaic in Southwest Sydney and Mosaic at Prince of Wales and ARIA at St George, into a researcher-ready data sets so that we can measure variation in practice in almost real time, and we can look at the effects of interventions. If we decide to pay people for following the guidelines or just give them feedback, we can tell whether that will work or not because we'll have a real-time measure of that variation. And one of the spin-offs of this is that we can then develop a near real-time clinical cancer registry with far fewer resources needed to maintain it and to publish it. Another spin-off is that we'll be able to identify clinical trial candidates, both for the feasibility studies and in actual practice as people come through the door. Next slide. So this is the rationale behind a project called CaVa, Cancer Clinical Variation. We put in to UNSW for some infrastructure funding a couple of years ago and were successful in getting a couple of 100,000. Unfortunately, due to COVID, they tried to reclaim some of that, but we have funded researcher for this year and part of next year to start developing the tools we need to join up the databases and to make them computable. So if we go on to the next slide, So Nazreen Kadan, who I think very many of you will know, is going to talk about the Southwest Sydney Cancer Registry, where it is and where we would like to take it, and what particular advantages CaVa has. So Nazreen, over to you.
Okay, thank you for the introduction, Michael. So I'm just going to talk about the Cancer Registry, where it was, and like Michael said, what we're hoping for with the CaVa project. So many will know the Clinical Cancer Registry program was established back in 2005 to supplement mandatory cancer notifications with disease stage, treatment, and quality of care indicators. We collected first course treatment for diagnosis up to 2015 and all course treatment after 2013. The amalgamation of the registries with the central registry provided a complete cancer patient journey in the one system and less duplication of effort across the state. And then the scope increased, like I said, to all course treatment, and that was achievable through automated capture of radiotherapy and chemotherapy data from the oncology information systems. But when the Cancer Institute seized their funding in 2019, we embraced this as an opportunity to build a local clinical cancer registry with some of the smarts that we previously had, but combined with the new technologies. And that's what Tim and Georgie will be presenting after me with the CaVa project. And the new vision for this registry is really to automate as many of the data collection processes as possible that were previously manual. We wanted to shift the focus of the team on data quality in the source systems and in the registry. We want data from multiple sources to provide the complete picture of the patient journey. We want to have access to data extracts and statistical tools for research. We want data visualization and tools and dashboards to monitor data quality and completeness, as well as to monitor service delivery activity. And we want to be able to assess compliance with guidelines, recommended therapy, and as Michael spoke about, unwanted variation in practice. New slide, Georgie, please. To have a comprehensive data set that captures the complete patient journey, Michael spoke about Mosaic, but we're hoping that the registry will encapsulate multiple sources for the different data elements that we need. We need to have accurate diagnosis and stage detail, or as accurate as we can get it, and pathology will actually be able to provide that in most cases for both public and private. We need treatment detail from our oncology information systems like Mosaic, but we also need some surgical data from HIE and the admitted patient data collection data sets. We need death data to understand our outcomes, so we're hoping to be able to link in with the registry of birth, deaths, and marriages. By combining and curating the data in an automated way, it's hoped that the outcome would be the researcher-ready data set, which is CaVa in this case, with high quality and complete data, and our manpower will then be focused on research projects and source system quality improvement initiatives. Next slide. So this is just an example of something we've worked on in 2020. So we focused on improving the diagnosis capture in Mosaic, and what we found since January to August, 42% of our cancer diagnosis had at least one error. But with the monitoring, the proportion of errors actually dropped from 54% in January down to 29% as it was in August. So we can see the impact that we've had straight away just by monitoring and feeding back to our clinicians. And up to August, we had 231 error notifications sent to our clinicians. 80% of those were actually resolved, and 20% of them were actually resolved in the same day, which was really good. And 54% were resolved within 5 days. The other audit that we've been running for a year and a half now is the mismatched care plan and diagnosis. So we're finding a very common error where the chemotherapy care plans attached to the wrong diagnosis. And so we're working on that with email communication with our clinicians to fix this. But this is an example of where the error in Mosaic cannot be fixed down the track and it'll remain. So this is an area that we're trying to focus on to get really quick turnaround time on the fixes. And we were at 12% of chemotherapy care plans were attached to wrong diagnosis. We're hoping to get down to 4%, and we're sitting at around we're sitting around halfway mark of about 9% at the moment. So this is an example of quality improvement work that we want to do, and it all feeds into improving the quality of the data that is attained and is available within the CaVa platform. So that's where we're at from my perspective. So I'll hand over to, I think Tim is next.
Thanks. Just before Tim starts, I was remiss. I should have asked everybody who has questions to put them in the Q&A, please. Tim, away you go.
Thanks, Michael and Nazreen. Yes, so as Nazreen said, we're building the CaVa platform very much on the back of and the experience of Nazreen's group running the clinical cancer registry in southwestern Sydney. And our aim is not to reduce a workforce or completely automate things, but rather refocus effort so that we can use the power of modern computing to do the boring clerical busy work and very much focus on the clinical aspects and the data quality aspects. So thinking about that and Michael's challenge of how do we actually get a handle on clinical variation in cancer treatment led us to a couple of functional requirements. First of all, as Michael's already said, we need to combine clinical cancer from various sources and EMRs. And there are two main ones used in New South Wales. That's the Electa Mosaic system and the Varian ARIA system. But around the world, there are many others, and indeed in the state, there are several others also used. It absolutely needs to be near real time. So Nazreen mentioned the clinical cancer registry. Despite the best efforts and funding and so on, such registries always tend to be a couple of years behind. And that really reduces the utility of these registries for fixing problems, as Nazreen said, on the fly or very shortly after they occur. So continuous reporting and analysis is really where we want to head. So the idea is not to have annual reports. Well, we can still have annual reports if you like, but they become much less important, but rather there's a whole series of online reports and dashboards which are always and automatically kept up to date so that people can actually look at the information when they need it or be prompted very, very frequently to keep up to date with the current state of things. And the rest we've already discussed about measuring effective interventions and trial candidates. Next slide, please. So the question is, how do we get there? How do we fulfill these functional requirements in the third decade of the 21st century? And I like saying that because that's where we are. We're now entering the third decade. We have to start to rethink things. And the fundamental principle that we're adhering to in the CaVa project is that absolutely everything needs to be computable. Next slide. So what do I mean by computability? Basically, it means that all the inputs that are needed to fulfill those requirements need to be in a form that permits algorithmic manipulation by computers. So by an algorithm, we mean a whole range of things, ranging from just routine reporting of data through to high-level statistical analyses, right through to highly complex and sophisticated machine learning prediction algorithms to detect potential unwanted variation. But the main thing is that the data needs to be in a form on which we can do algorithmic manipulation. And that means not just having data in some sort of electronic form, but rather having it represented in a syntactically and semantically consistent form. So what do we mean by that? Syntactic consistency means having the same structure, that is, data in the same tables and fields and columns and so on, across all of the different sources. And semantic consistency means that the ideas and concepts contained in those data, in those tables and fields and columns, are represented in the same way. So a trivial example is that male and female is always coded the same way as, say, 1 or 2 or M and F and so on and so forth. That's a completely trivial example. But you can imagine that very quickly it becomes quite complex, and it's very, very important that we transform data into a semantically consistent form so that we can actually compare exactly like concepts from different source systems. Which naturally leads us to the next section, but before we go there, perhaps we need to ask ourselves at this juncture a question, and that is, has the traditional cancer registry model broken? And Georgie pressed the forward button a few seconds too early. Never mind. But we don't. We think that at this juncture we need to rethink the way traditional cancer registries and indeed all clinical registries operate. And that leads us naturally onto Georgie's section.
All right, Tim. I was excited about the animation. So as Michael will get set in his overview, I'm going to talk to you about how we actually do that, how we're going to get the data that's sitting in the Mosaic database into a form that meets the requirements of computability that Tim just described. And the first thing I'm going to do, I've split it into two parts. So the first bit I'm going to address is sort of the conceptual overview. So what's the overall approach to tackle this problem? Before we try and give some insight into what practical steps we actually have to take to be able to achieve that plan. So the first thing is every application that we use has its own data model, which means that sort of the database structure that's used to store the data that's been input by the users. And so we can't even really assume, like I know Tim said, there's some main system. So we're looking at Mosaic and ARIA. Even in each Mosaic implementation, we can't totally assume that each instance has the same underlying data model. So in practice, each instance of that will have customizations to the application itself, but also there are sort of local operating procedures that affect the ways in which people use the applications, that also affects not only where each data point is when you go and look for it in the database, but also the way in which you can interpret those data points and what they actually mean in terms of clinical evidence. And so if your goal is to centralize data from multiple systems, and we're talking about all of those different systems Nazreen mentioned, one robust way of doing this is to implement a common data model. And I think if we have any Power BI users in the audience, you might be familiar with that concept, but just generally, it means we define a schema that holds data from multiple source applications, but also that this schema has to have sufficient expressiveness to suit the target analyses. And so in this kind of context, what we mean by schema is the structure of the database itself. So that's the tables, the relationships between those tables, and then the data types, for example. And by expressiveness, we mean sort of the trade-off between how prescriptive the schema becomes. So how much detail can we capture about each clinical event and the context in which it was captured, but also the trade-off between complexity and the richness that we get about each concept. And so the way that we do that is by harmonizing concepts, relationships, and vocabulary. So that's what Tim was talking about with semantic and structural compatibility, but also its interpretation. And so we're not just incorporating the knowledge about the data, but also our knowledge of how this data provides the best available evidence of what's actually occurred. So hopefully the case for using a CDM is fairly obvious, because it allows us to represent detailed clinical care data that's been derived from multiple systems in a rigorous way. But we also should note another important reason for doing this, which is that if our data source adheres to an accepted common data model, any analyses that we develop in our system can be run against any other system that also adheres to the same common data model, which greatly improves efficiency and also research reproducibility. So I don't know if you want to imagine being able to run your analysis on somebody else's data without having to go through the administrative headache of actually getting access to the data, because you have a defined set of assumptions about what the data will look like. So obviously, we don't want to be reinventing the wheel. So thankfully, there's a mature community internationally that's been thinking about this problem in this particular context for quite a long time. So the group we're talking about is Odyssey, which stands for the Observational Health Data Science and Informatics Group. And they published the OMOP CDM. And so you can see down here, if I put a link, I don't know if we'll send the slides around later, but that's the Observational Medical Outcomes Partnership. And the core goal of their model is to capture data from both clinical and administrative records and provide a robust methodology of generating evidence that you can use to support your research. And I think it comes up quite a lot when you're looking at the benefits of the OMOP model, but the concept of generating evidence is really, really important. and it explains some of the conventions and assumptions around the model, and it becomes relevant later when I go through some of the oncology applications. But when you're bringing data in from multiple sources, you kind of naturally expect that there will be areas of conflict. No data source is perfect. And so you may have contradicting evidence, you may have missing in one system. And so OMOP has a system of handling this. And the convention is to search for whatever data point is considered by convention to be the best available evidence for any particular event occurring. And so a simple example is for the best evidence for a patient having actually ingested a drug. So the best evidence of that is a medication administration record. But in a lot of settings, you don't have that. So in an outpatient setting, and even in a lot of inpatient settings, you won't have the actual administration record. You will only have perhaps the pharmacy dispensing record. So that's at least evidence that they received the medication. It's not necessarily evidence of when exactly they ingested the evidence of the drug itself, but it's evidence that the patient got it. If neither of those exists, the next best evidence of medication administration is, of course, a drug ordering record. So in that kind of level of hierarchy, you can see the focus is on getting the best available evidence for any clinical event. So the OMOP CDM itself, which is this picture over here on the right, is much too complicated to go through in a valuable way in this session. But I've pulled out a few motivating examples that will hopefully explain the fit of this CDM for the goal of what we're trying to do. So I'll go through the way that they handle concepts and vocabularies, and also specifically relevant in the oncology context is their diagnostic model and the episodes of care and the way in which they treat computable regimens. So this here is an example of an existing vocabulary that exists in OMOP. So what they have done is they've ingested a lot of the main international vocabulary. So in this instance, it was ICD-10 is already available in their data structure. It has a defined concept ID, and it's got defined relationships by which you can get a lot of granularity into these concepts and the relationships with other concepts. So you can see here on the right hand, the way in which the relationship is used to be able to translate between ICD-10 and in this instance, SNOMED. So that's really useful, and that is often quite a tedious step in the first kind of phases of clinical research is to harmonize that. And so that's already there if you're operating under these kinds of vocabularies. But really importantly, I think, and there's a lot of value in this subsumes relationship here. So again, if we go back to the best available evidence, it's quite possible that if we have this increasing level of specificity, we can then we can define our research question or our cohort builder at these different levels and look for equivalencies. So say you're looking for some reason to grab all of the patients who have been assigned, who have experienced myeloid leukemia, but then you can use the relationship hierarchy to grab everybody who's been assigned these lower level, more specific diagnoses. And so that gives you a lot of opportunities there to look at equivalencies between the terms. Also importantly, we'll look at an example of where you're able to import custom vocabularies. So what we've done here, this is the portion of the CDM that describes concepts and their relationships to each other. So in this instance, what's important to notice is that the concept relationship that I looked at from Athena is over here, but we also have the concept synonym. So already we can look at, on the synonym table, we can use translations in different international versions for direct equivalencies where it's just a string change, but the code is the same. And then the concept relationship is used when you're mapping concepts between vocabularies or within a vocabulary. So here's an example from importing the MBS. So that's not one of the core vocabularies that exists within OMOP because it's obviously Australian specific. And so we've imported that, and we've imported the relationships that come from the MBS chapters, sections and subsections to be able to leverage those relationships By having the strict definition of how one should import new concepts, we can already start to use some of those traversals against the MBS, which is what our source data has. Also really important, I think, is that when we're defining oncology cohorts, it's very complex. We're not just looking for the diagnostic information. Most observational data sources don't support looking at the morphology, the metastases, biomarkers, disease staging and grading. And also, this is really, really key in the oncology working group, is that even though you have, say, all your pathology results and you've got a whole bunch of relevant observations, they're present, but they're not linked back to which diagnosis they refer to. And because you can't do that linkage, you can't group those. If there's multiple cancers, or if there's pathologies that that exist for different reasons, you cannot make any relationships between them, whereas the OMOP CDM does actually support that. And also, when we're looking for people who have been assigned to treatment regimens, they're significantly more complex than just a straightforward prescription. We're looking at repeating structures and relationships that are really important to defining the cohorts. And so the way that OMOP supports that is by, it uses the cancer diagnosis. The core diagnosis is up here, the carcinoma of the breast in this example. But by having the relational model, you can assign the morphology and the site via these relationships, has morphology and has an anatomic site. And then further down, you can relate the grade and the tumor size to the specific diagnosis that they refer, rather than just having all of those disparate observations that exist, but perhaps aren't actually linked back to that exact diagnosis. So, and the other also key conceptual issue that hasn't been available, I think, in a lot of such systems previously, is the ability to group diseases into disease episodes. And so OMOP allows you to group episodes that describe the disease extent. They also describe the dynamic disease. So that's all the observations that occur under a disease, and also the overarching disease episode. And I will give you a worked example of that later, because I'm not sure that is immediately obvious. One thing also as well is that we're looking at computable regimens. So core to the concept of CaVa is one of the things, if you are looking for variation, you need to know whether the treatment that was given was the treatment that was intended. And in order to understand the treatment that was intended, you must have computable regimens, which has to date not been available. So there is some existing work in this space, which is the HemOnc regimens, which have been converted to OMOP. So you can see here an example that they've got a whole lot of modifiers and relationships to do with the drugs that are assigned to this regimen, and the regimen classifications, which are very useful and expressive and give you a lot of information about the regimen. But the one thing that they don't do in terms of the core remit of CaVa itself is they don't give you a treatment schedule. So core to the OMOP CDM is that they really look at observational data, and what that means to them is that they look at things that have actually happened. And so up until this point, it hasn't included the facility to really, like, a prescription is something that has not yet happened unless it's being used as evidence of a drug ingestion. But we wanted to kind of change that paradigm and include the prescription as an observation in and of itself, because the fact that a treatment is assigned is core to that. And so we have proposed an extension to the model that will allow us to capture information about the regimens, but also what that actually means in terms of, we expect them to be getting this drug on these days. Did they get that drug on those days? And what's the tolerance for change in between what's expected and what's actually delivered? And that's going to really help us to understand the treatment variation concept. And here's the worked example of an episode. So here we have the base diagnosis example. So here we have a base diagnosis of carcinoma of bladder, and to that we can apply the modifiers of topography, stage, genomic variant, and the fact that there has been disease metastases. And if you want to go later again in more detail, you can. There's a lot more information at this link down here. The way that you split this this episode into, sorry, this disease occurrence into an episode is we define a parent episode. There's a hierarchy of episodes where you can have one overarching episode under which you link multiple different child episodes. And here we look at the disease extent, where each disease extent, this is like a one-way valve. At the beginning, everything is confined disease until there is evidence of invasive disease. So you kind of, you move through this progression from confined disease to invasive disease to metastatic disease from left to right, and you use the first available evidence to trigger the move from one stage to the next. And so every diagnostic episode for cancer will have a disease extent episodes, and whether it has one or more of these will be based on what evidence there is in the observational data. And each disease extent episode has a defined start date, which is the first available evidence, and it has an end date, which is either the date that it moves on to the next disease episode, or the last available date of any evidence at all, so kind of the current, or death date, obviously, as well. So that's the disease extent model. Under that same episode of care, we have two types of, or two flavors of episodes underneath. So we saw that we had the disease extent, and then we compare that to the dynamic disease progression here. So that's more the kind of the activities that occurred within that disease progression. And you can see here as well, again, we go back to the concept of looking for the best available evidence. If you follow down the bottom, we've got progression, remission, progression, complete remission, progression, remission. That doesn't mean that there has been a record in the source data that says disease progression. It means that we infer disease progression from the initial diagnosis. We infer progression, even though perhaps there's no available pathology from which you can infer it. But the fact that they had to have a follow-up cystectomy at the end there, you can infer between the two periods of remission over on the right-hand side, there must have been a progression. You can insert that episode, but you don't have any additional data about it. And so from that, that fills out the disease episode through the dynamic events that have occurred. And so then I'm moving on to the practical considerations, so the more kind of the technological side of things. This here is a sort of a more fleshed out example of the conceptual model that Nazreen spoke to. So the point of view, if we move it down sort of further downstream, we're actually thinking about the building blocks of what's being required to produce a researcher ready platform. And so what this actually is, it's a fairly straightforward ETL process. So ETL is extract, transform, and load, which is quite a common database operation. It's a mature kind of process, but we're trying to sort of provide the best practice implementation of that. One thing to note here is that eventually the de-identification step will be on site at the source, but at the moment in the development phase, that has to be within the hosted platform itself. So we look up here the data sources as described. There's numerous, and then the CaVa application itself is the sort of the lighter blue in the middle, where we have a pipeline model that takes the input data and transforms it to the target CDM. And on top of that, we kind of have an overlay of tools. So just a browser that can you can look through the data, explore it in a sort of an intuitive fashion. And then we're going to have a suite of analytic tools and libraries that make it a lot easier to interact with the data. And then also some simple applications such as a cohort builder that allow you to specify your cohorts and stuff in a fashion that doesn't rely on you having sort of particular programming experience. And then we pull into that the external references and standards that that have been referred to already. And then, those can be published for all of those end use cases, which is, quality and safety, research, either primary or secondary research, and then also, registry functionality. In terms of architecture, the requirements that we've got there are that it is obviously secure. At this point, it's centralized. We also want it to be really modular, so we don't want to be redeveloping something every time it has to be changed. Initially, I wrote low cost, but then I realized it's more accurate to say we don't want prohibitive costs on that. Anything that's going to meet these requirements is going to have some cost with it, but we're trying to build it on open source requirements and have things that don't make cost the main reason this can't be done. And don't try and ingest this diagram, but this is the Erica platform that has been developed for this. So this Erica system platform has met an incredibly high bar for security. So it's been approved by the eHealth New South Wales PSAF in 2019, and it's also recently been moved through to the approvals for the Australian, so the national level linked data. So it's been approved for both of those, but it doesn't really rely on proprietary technology. So wherever possible, we've created an open source stack and made it configurable. So the configurability means that you can specify, you can scale up and scale down the performance of this. So it should be quote unquote easy to deploy within the LHDs or other organizations. Some of the challenges around this is that traditionally ETL, which is again the extract, transform, load, traditionally is hard coded. So it's a lot of very specific code, grabbing this particular data point, putting it in this particular point. It's not well suited to systems that change or that get used differently in different contexts, and it's really costly to maintain because of this inflexibility. And you get a really limited ability to blend data sets. So you have to, if I want to share my data and somebody else wants to share theirs, basically we need to develop the ETL from scratch for each of those systems. We have tried to get around that using a low-code ETL. Unfortunately, we can't go sort of a no-code approach, which I don't know if people have seen systems where you can kind of drag and drop or use like a graphical interface to specify where the data comes from. The mappings for those disease episodes are obviously too complex to not have any code. But where possible, we use a system that will generate the software code components based on configuration files. So those configuration files can be understood and read by clinical analysts, not just the technical data analysts. And wherever possible as well, we want to generate those configuration files from the system settings. So that means that I don't have to sit down and write a new configuration file for a new system. I can just query the source database to try and generate that system sort of to plug and play where we can. Similarly, we also want to look at data-driven transformations, not necessarily code-driven. So that's where you can teach the system. Here's a few examples. I'll work through these manual examples, teach the system how to do it, and then it will be able to use machine learning to do that transformation. And importantly, the whole system is self-documenting. So by that, I mean that all of the documentation is generated from these configuration files and from the code itself, which means that we're not having to chase our tails redocumenting the system for each source system, which gives us a lot more power when we are trying to do quality assurance. In terms of status, the mature systems, so Nazreen and her team have given us the Mosaic extract, which is great, from Southwest Sydney. We have generated the object relational mappings for both the source and target data models. And we have a fairly robust pipeline for doing this mapping of the simple concepts. And we've also proposed the extension to OMOP, as I described before. Some of the things that are in development, so the more complex clinical concepts are underway. At the moment, we've got sort of the initial diagnostic model, but looking at grouping those into episodes is going to be an iterative process working with clinical end users. We're looking at increasing how expressive the generation can be, including those machine learning steps that I described before as well. Tim will go through in a very brief moment some of the initial data exploration tools that have been worked out and the validation pipeline as well. We're still working on also the incremental load functionality. Some of the things that I have on the horizon but haven't been started, looking at extracting concepts from free text. That's a non-trivial task. There's going to be a lot of steps in that, but it will greatly increase the power of the system. And also look at really maturing the data exploration functionality. And then core to this entire premise is pulling in those additional data sources. So I'm going to hand over to Tim now, and I'll hand over in terms of speaking, but also in terms of actually presenting. So I'm going to stop sharing for a second. Okay, there you go, Tim.
Okay, thanks, Georgie. So I think Georgie was a bit too modest. She left out a few things that have also been done in the last a couple of months. So she mentioned EverQ and Ingham. What Georgie didn't mention is that she's developed machine learning models to completely parse both of those resources, which are represented as sets of web pages through thousands and thousands of web pages. And she's developed a model that will parse that, so automatically read all those web pages and put them into the database computable form that she was describing, which I think is in itself a major step forward, and caused the jaws to drop somewhat at the Cancer Institute and also the Ingham people in the US. But what I want to talk about very, very quickly, because we are running out of time, is analyzing OMOP data, and this is very much work that's we're just getting started on, and making it easier. So as Georgie alluded to, OMOP has a dirty little secret, and that is that it's all fairly complex, and it trades off ease of use and simplicity for these syntactic and semantic harmony that we were describing at the beginning of this presentation. But it's not that bad, and typically experienced data analysts can get to grips with it in a day or so, and it is extremely well documented. There's a whole book called the Book of Odyssey, not written by Homer, but it's some 300 or 400 pages of very good documentation around the whole of the OMOP common data model and concept system and so on and so forth. But nonetheless, it's still intimidating for occasional analysts, such as many clinician researchers, and it does assume very good familiarity with SQL, which is not something that every health researcher has a lot of familiarity with. So let's have a very quick look, and I mean a quick look at a simple analysis of OMOP data, and this isn't a cancer-specific one. It actually comes from the Book of Odyssey. And I'm not going to go through this in detail, but just imagine that we want to look at, say, the rate of angioedema in the first week following introduction of an ACE inhibitor, stratified by age and sex. So we need to do a couple of things. We need to define the exposure when they were first exposed to the drug, an outcome definition, meaning occurrence of angioedema, and epidemiological concepts such as the time at risk, so we can compute the incidence rate correctly in the first week following treatment. So let's have a look at some code as provided from the Odyssey group in the book. So this is the traditional way of analysing OMOP data. And now the font here is deliberately small because I don't want you to read all of this, but this is to give an impression of the amount of code needed, which is not a huge amount, but it's somewhat complex, and the fact that it's all written in SQL. Which, once you're familiar with it, starts to make sense, but can be a little bit hard to get one's head around. So you have to do all of those steps and some more steps, and then finally we can actually visualize the results in terms of age-specific incidence rates broken down by sex. So that in itself is not that bad, but we think that we can do better than that. So one of the sub-aims of the CaVa project is to try to make OMOP easier to use, and specifically for oncology data, but not just for oncology data. Further than that. So where we will be using meta-programming in the R open source analysis system and ORM, so object relational mappings in the Python open source ecosystem that Georgie mentioned, to make working with the OMOP data, we think, very much easier. But on the other hand, also without limiting generality or of what analysts can do. So we don't want to restrict what people can do, just make it easier. And I'm collaborating with Kirill Merler in Zurich, who's at ETH and Syncra. And that's one of the advantages of using open source software stacks, is that you get to work with really smart people all around the world at almost a moment's notice. And it's very productive. So what we're doing for the R ecosystem is defining an abstract data model over the top of the OMOP database, so that analysts don't have to deal with all of the nitty-gritty and all the 6 million tables and so on and so forth. So this all works with the R ecosystem called the Tidyverse, which is a very, very nice system for manipulating data. And it allows you to write data manipulation in almost English-like pipelines. So it's not English, but it's much easier to understand than writing lots of SQL or other things. And as we go along, we'll gradually be adding some higher level helper verbs for, specific for the OMOP translations. So here's an example. So let's just look at the numbers of females and males in the CaVa data. So this is actually running against data which has been extracted by the Nazarene from the Southwestern Sydney Mosaic clinical system. It's just 3 years of data. We haven't validated any of this, but this is actually transformed by the ETL pipeline that Georgie described into an OMOP formatted database, and then we're using R to read from that. So you can see there's just a few lines of code there to look at the relative proportions of males and females in the Mosaic data. Now, okay, you might say, Tim, we're not going to get a Nobel Prize for that. All right. So let's look at a slightly more ambitious example. So let's look at, say, for instance, looking at all skin cancers. So there are many, many ways of actually pulling out that cohort. A very simple way would just to be looking for the occurrence of skin in the description of the condition, which is what I've done here. A few lines here, which we will simplify further in future versions of this framework. And we can draw a bar chart broken down by males and females for the various types of skin cancer treated in southwestern Sydney. And you can see most of them are unspecified malignant neoplasms of the skin, on the face. And the main difference between males and females is that the number of males treated for malignant melanoma is much higher than females. That in itself is an interesting clinical fact, which maybe no one's actually ever seen before from the Southwestern Sydney data. But we're able to write that in just a few lines of code, and it runs in 10 or 15 seconds. And so that's where we want to go. Another example. So here, let's look at the age distribution of people treated for breast, colon, and cancers and lymphoma. So again, there's the code on the left, and we can produce sort of comparative age distribution. So this is directly from the CaVa OMOP database, which in turn is directly derived from the Mosaic data. And the aim is that we will be reloading these data sets once a week. So we'll be looking at data that's no less than a week, no more than a week old. You can see in these age distributions what you sort of expect. Breast cancer at the top, sorry for the small labels. Colon cancers in the middle, and lymphomas at the bottom. The colon cancers overall, mean age is slightly older. And lymphomas, you can see here, a typical smaller peak in the young adult age groups, which is presumably the Hodgkin lymphomas. So that again in itself is an interesting teaching point, and it's derived directly from our clinical data. And a final example, you can't look at cancer data without looking at Kaplan-Meier curves. So here's again a small number of lines of code. It's a little bit in the middle is a little bit complex, and one of the aims over the next several months will be to simplify this further. But we can run that code, and here we have, again, we're looking at lymphomas. So we pull out all of the lymphomas treated in the last 3 years, looking at the survival probability. So these are Kaplan-Meier curves for males and females with confidence limits. And this is proper sensor data, so you can see the declining number of risk with the lower follow-up as time goes on, and so on. And you can see that the survivorship of males is significantly less than females. I wouldn't put any store up by that. This is unvalidated data. It's only for the last 3 years. So this, I'm not drawing attention. This is an actual bit of research outcome, just merely the capability of being able to actually look at these sorts of analyses with very, very little effort. And of course, we can then go further and start to fit statistical models. So here's a proportional hazards regression model by gender on exactly the same data set with just a few more lines of code. So the idea is that we can use this programmatic analysis framework not just to write code per se, but it forms the basis of creating dashboards, quite complex reports, including, as I mentioned before, getting rid of annual reports and having continuously updated reports of the same length, typically hundreds of pages, but automatically updated every week. Interactive applications such as cohort builders, so you can extract data for analysis in other systems, and of course for writing scientific papers. So just changing tack a little bit, we haven't spoken about the governance of this project, but we should just mention that it's completely under the oversight of South Western Sydney LHD HREC, Ethics Committee, and currently we have approval to develop the data infrastructure. But specific research projects will still require separate HREC applications to use the CaVa data and framework, but we think that those applications should be regarded as low or negligible risk applications, mainly because we've put a lot of attention into addressing the 5 safes framework. We don't have time to talk about this in detail. You might have heard of it before, but the 5 safes framework describes a whole mechanism or framework for safely dealing with these sorts of data that are assembled and then used on a routine and ongoing basis. So it's all around safe projects, and that's where the ethics oversight is still required, but a lightweight process of LNR applications. Safe people, meaning that we pre-qualify researchers, and the ERICA environment that Georgina mentioned that we're using already has training, online training, including exams and so on, to make sure that researchers are aware of their responsibilities and how to keep data safe and so on. Safe data, Georgie mentioned that we're putting the data through a process that makes sure that we remove all potential identifying information. Safe settings, the ERICA environment itself keeps the data very safe. And safe outputs, again, we train researchers in looking at the outputs to make sure that there's no leakage of private information in the results. So we're attending to all of these things, and that's why we think an LNR approach is probably going to be feasible. So lastly, where are we going with all of this over the next 12 months or so? So the short term, in the first quarter, we're going to complete acquisition of the Sydney Mosaic, Southwestern Sydney Mosaic data, doing more work on the protocols and regimens, and more work on the analytics framework. But we're also applying for various bits of funding, including a CRCP grant in conjunction with, we hope, a large private sector cancer treatment provider, and applying for further HREC clearances for extensions to variant ARIA data at St George Hospital, and also specific bits of surgical data from the Cerner EMR death data that Nazreen mentioned, and then potentially down the track radiomics data in conjunction with Lois Holloway's group in medical physics and the whole AusCat project, which really have a much better handle on radiomics data. And then further on next year, doing the work with St George Hospital on the ARIA data, applying for an HNNRC partnership grant in conjunction with the Cancer Institute. As I said, we have been very impressed with Georgie's work on making their EverQ protocols computable, and then starting to actually then leverage all of this infrastructure we're building to provide new reporting systems for South Western Sydney, and of course other areas and organisations that might use this. And then later on, finally addressing the challenge that Michael said right at the beginning, later on next year, I should say, in developing machine learning methods for actually detecting and characterizing unwarranted variation. And most importantly, developing a sustainable consortium-based business model, because these things do cost money. We can't rely on research funding for this sort of infrastructure funding. So we're very much looking at a consortium-based model, because we think many other organisations will be interested in this work and will be willing to contribute to it. So finally, what's the big picture in all of this? So we're not just creating a new clinical cancer registry to support cancer services research and quality assurance in southwestern Sydney LHD. So yes, we are absolutely doing that, but not only that. What we're also doing is creating a framework and a technology for creating such new cancer clinical registries to support cancer services research and quality assurance in many locations and organisations. So we're not just doing it specifically for southwestern Sydney, but we're doing it in a generic way that can be reused to create the same things in other LHDs, in other organisations, in the private sector, as well as the public sector, and also internationally, because people everywhere are interested in these things. But the main thing about this framework and the technologies we're using is that we think the next generation, because we're focusing on automation everywhere, leveraging the very latest machine learning methods, and using the very best in software engineering practices in terms of using open source tools and the latest what are called DevOps methods for doing this on a continuous and sustainable basis with low maintenance. And we think that taken all together, that's quite a unique proposition. And that this is actually world-class research, which will have immediate benefits locally, but will be of interest nationally and internationally. And we're very excited to be able to continue working on this in 2021. So that's the end of our presentation. I think we may have just a few minutes left. We're only gone slightly over time for questions.
Congratulations, Tim. I didn't think you would make the 53 slides in 53 minutes.
One slide per minute.
One slide per minute. If people have got questions, I'm not sure that you can unmute, but you can put them in the Q&A or in the chat. We've just got time for a couple of questions. I might start by asking you the curly question of what do you think will be the first win with this? Where is it going to make a difference first?
I think we'll be able to work with Nazreen to provide some really detailed and really highly targeted data quality reports, which will be able to be automatically generated once a week and sent to people. And I think that will, as Nazreen already showed in her slides, have an immediate impact in terms of data quality. But also, I think that just the descriptive epidemiology of what's going on in the southwestern Sydney cancer services, I just showed those age distribution graphs. I found them fascinating. They're probably not surprising to cancer clinicians, but I think it's incredibly valuable that people can actually see the epidemiology of the services that they are actually providing. And I think we'll be able to start to provide that quite quickly next year. But beyond that, the sky's the limit. So I think this will provide a basis for lots and lots of scientific papers, but also lots and lots of very sophisticated reporting systems around the outcomes of cancer treatment. And all of that underlying our ability to start to work towards developing routine systems for detecting cancer variation in treatment and outcomes.
So there's a comment in the chat from Shalini saying that lung cancer puts its hand up for immediate quality reports. Who's doing this on a 6-monthly retrospective basis. I guess that raises the question of sustainability, user costs, and the whole business model for getting access to the data set. Got any thoughts on this based on, say, the Erica model?
Yes. So Erica is actually a joint production between the Centre for Big Data Research and Health, which is part of UNSW Medicine, and South Western Sydney Clinical School, in that I've been very much involved in the Erica development at the clinical school, and the Ingham Institute. So Erica is a joint production of South Western Sydney Clinical School and the Ingham Institute. And it's been very successful in that we developed it for use just within UNSW medicine, but then other people said, oh, that's very useful. And then the Australian Institute of Health and Welfare said, we'd like a version of that. So we actually developed a partnership or collaborative agreement with them, in which they started to co-fund its ongoing development. And then the various other organisations said they're interested. We applied for ARDC funding, and last year we got nearly $1 million in funding from ARDC to further develop Erica. So by the end of next year, there'll be a consortium of 8 players, including University of Melbourne, UNSW, University of Melbourne, AIHW, University of Western Australia, and several others, all contributing funds on an ongoing basis. And the funds are not huge, they're $50,000 or $100,000 a year to the ongoing maintenance of and development of the Erica software platform. So each of these organizations run their own instance. We don't run it for them, but everyone is sharing in the development. And we think that sort of consortium model is potentially highly applicable to CaVa, or the frameworks that we're developing, in that each organization, we don't have to have a single pooled, centralized CaVa database, which starts to get very expensive to run, but rather many organizations, each LHD, private sector and so on, can run their own, but they all share in the development of the underlying technology through this sort of consortium arrangement. So that's what we'll be pushing.
Great. Okay, so we are now out of time. Sorry, Georgie, did you want to say something?
I was just going to add something really small that speaks to kind of both of those comments, I guess, which is that I am very open to receiving use cases like the lung cancer reports that have been mentioned, because exactly like you said, Michael, in terms of realizing value, we want some really compelling use cases that really make this work for users like they will use it, like they want to use it. So if you do have particular use cases where you can see, oh my gosh, this fits exactly what I want, we can't, guarantee that we'll start working on those immediately, but they really, at this point in the program, it's the time to sort of influence the development direction. So very open to receiving those from anybody who has something like that. Okay,
be careful what you wish for. On that very generous note on Georgie's part, I'd like to thank Tim Churches, Georgina Kennedy, Nazreen Kadan for a very stimulating presentation. And we hope to present this many times in the future to show the progress that it makes and the influence that it has. So thank you very much, everyone.
Thank you.